Well, as we said before, today's speakers are Dr. Alynn Martin, who is an assistant professor at the Caesar Kleberg Wildlife Research Institute of Texas A&M University-Kingsville. She is a disease ecologist and epidemiologist, and her work focuses on disease transmission dynamics and applied methods for mitigating disease impacts on wildlife. Dr. Sandra Rideout-Hanzak is a professor at the Caesar Kleberg Wildlife Research Institute, Texas A&M University-Kingsville as well. She's a fire ecologist and restoration ecologist, and her research focuses on fire impacts on rangeland and habitat quality and how a season of burning affects ecological outcomes. We'll give them more opportunity to introduce themselves in just a minute. We do have poll questions. Why is it doing this? Ryan Anderson, are you able to launch the poll questions? It's giving me some sort of error.
Yeah, I will do it.
Thank you.
It's the May, right?
Yep, we should have a May pre-question and a May host question. We'll give that a minute. While we're starting that, did your student end up making it to the webinar today?
I believe so. Rachel, I think I saw you on here. Are you on?
Yeah,
yes.
Hi.
Hi, Rachel.
So if you guys want to take a minute and start to introduce yourselves and Rachel, that would be great.
I've got a little introduction slide. Do you want us to go ahead and do that now, or?
Sure. Yep. I can stop sharing and we'll transfer over here.
Let me go ahead and get my screen share on here. Okay. And start. And swap this. Okay. All right. Are y'all seeing my normal presentation or my notes?
Normal presentation. Looks good.
Awesome. So thanks everybody for jumping on to our presentation here. We're going to do sort of a co-presenting situation with Dr. Rideout-Hanzak and myself, talking about our project in South Texas, looking at the effect of prescribed fires on controlling tick abundance, and hopefully we'll dive in eventually in this project to actually pathogen carriage control, but we're not quite there yet. So I just want to preface that these are very preliminary results we're going to be talking about today, so I don't think we're going to take the full 50 minutes, but we're hoping for some discussion at the end, and if anyone's got some ideas, we'd be more than open to hearing those. So real quick, if I can get my slides to change. I just want to introduce the team. So first we've got Dr. Sandra Rideout-Hanzak. She is our burn boss. She is very humble, but she has burned in many different landscapes in the pine forests and coastal plains in several southeastern states. She's burned in mixed hardwoods in the Appalachian and Piedmont regions, and then the high plains, rolling plains, and southern coastal grasslands. She's a senior wildland fire ecologist and a licensed lead burn instructor for the Texas Prescribed Burn Manager Program. Myself, I'm a disease ecologist. I mostly work in medium to large mammals, but I started in tick work when I was a postdoc with the U.S. Geological Survey. I actually worked in the Appalachian region on a project looking at control methods for Lyme disease and black-legged ticks and whether or not deer reduction strategies were effective for that. And then we've got a fantastic student on this project, Rachel Walters. She started with us in January of 2024. So she's got one semester under her belt. She's newly here, but she's taken the project and run with it. She comes to us from Allegheny College in Pennsylvania, and her background is looking at diseases as well. She studied fluke parasitism in snails, and she also was involved in a plague project with the U.S. Geological Survey and National Park Service when she did some tech work with them. And so just a little bit about our institute. So we are with the Caesar Kleberg Wildlife Research Institute, which is a nonprofit organization and one of the leading research institutes in Texas. Our institute is nestled within Texas A&M University-Kingsville, and we are based in Kingsville, Texas, which is South Texas. So we've got the little red pin there. If you're familiar with Corpus Christi, we're just about 40 minutes southwest of there. And so the impetus for this project is that Dr. Rideout and I have, well, general interest in tick control methods, but also understanding the human and wildlife disease portion of it. And when we were getting into the literature, when we first started this project and we wanted to explore, like, can we use fire as a control method? We quickly found out that there are a ton of dynamics that need to be considered when looking at this question. And I've listed a few here. We're not going to go through all of these, but just things that we've been thinking about in our own project is the tick source that the papers might be using. So there's a lot of variation when you're collecting ticks from the environment versus off of a host, and how to interpret those types of data post prescribed fire. Also, the return interval of the fire. Some papers have looked at short return interval, so 1-2 years in between prescribed burns. Some have looked at longer return intervals up to 5 years, and it's been hard to compare cross studies because of these differences in return interval. And then also the burn plot size. We've started to conduct a review on what's out there in the literature with prescribed fire with regards to tick control. And what we're finding is in the literature, a lot of times the burn plot size is actually just missing. And maybe this is a wildfire situation where it's not well documented. Maybe they're really small patch burns and it's a patch burn within a ranch and the ranch size is reported, but not the actual burn size. And so we're very cognizant of that, and we were interested in looking at that as well within our study design, although our burn plots have a range, and we'll get to that in a second. But overall, one of the major gaps we saw, and that we won't be talking about today because we haven't gotten to this quite yet, but that pathogen carriage is often not assessed following prescribed fire. So a lot of folks are looking at, do we see decreased tick densities post-prescribed fire? Do we see decreased tick carriage on hosts? But often we're not looking at, they're not exploring pathogen carriage. So there's a couple different dynamics we might see here in that, post-prescribed fire, ticks are forced to get on host because the environment is no longer suitable. And so does that increase the potential of them being exposed to pathogens and becoming infected with pathogens? And then if the overall result of prescribed fire is reduction in tick density, even if you've reduced the density, is it a higher likelihood that if you come into a contact with a tick that it's actually going to be carrying a pathogen? So these are all the types of questions that we've been thinking about in our research system. This is not meant to be an overwhelming table that we actually go through all together. I just wanted to list the common species of tick that we find in South Texas. There's at least 10 hard tick species present here. Some of them are 3 host ticks, so there's a little superscript of a 3. Some of them are 1 host ticks. I've got an asterisk there for the ones that are, or sorry, a plus sign for the ones that are primarily involved with human tick bites. And then I've got an asterisk for the cattle fever ticks, which are primarily, or one of the most important, or the 2 most important tick species for our agricultural systems down here, and I'll get into that in just a second. But what I just want to demonstrate is that all of these species of tick are capable of carrying either human disease or human disease causative agents or animal disease causative agents. So there's a lot to be learned from sampling all ticks in this system and looking at what they're carrying. And so one of the reasons that we're super excited about this project is because not a lot has been done with prescribed fire in general, but especially in Texas. I think there was one paper we found that came out of Texas, but it was from a park region. I think it was outside of Houston, and in the discussion, Rachel can correct me if I mix that up. But we're down here in South Texas, and we have some interesting tick dynamics happening here. So the most important is for our agricultural system. We've got these two species of cattle fever tick. These are Rhipicephalus genus ticks, and they're capable of carrying Babesia, which can spread into our agricultural animals, specifically our cattle, and they're a one-host tick. They have been a problem in the US in the past. There was an extensive cattle fever tick eradication program that worked throughout the southern United States, started in about the early 1900s, I think it was 1906. And the program involved 14 states altogether. Actually might have been 15 if you include California, because that's not truly a southern state. And for a long time, they worked to dip cows to get rid of these two one-host species ticks. And cattle fever tick was technically declared eradicated in the US in the 1940s. However, the cattle fever ticks are still very common in Mexico, and there's been some evidence that they're developing acaricide resistance. And so there's still a problem in South Texas with the movement of both agricultural animals, but also wildlife, which doesn't respect national borders. We still have zones where there are surveillance areas and infested premises, and that's what this Texas fever tick quarantine area is showing. The surveillance areas are those yellow zones, and the infested premises are in red there. And this cattle fever tick system is particularly difficult because of the wildlife component. And so in South Texas, in addition to the ticks feeding on our cattle, they're also parasitizing our wildlife. So white-tailed deer is probably a familiar species for folks on this call, but you might not be familiar with this animal on the left. This is the nilgai antelope. This is an invasive ungulate that is native to India. They were introduced in the 1920s to 30s on private ranch lands, and they have flourished in the South Texas climate. They're known to be carriers of both the Rhipicephalus ticks and Babesia. And so between these two species and cattle, the tick can be maintained in this system in South Texas. Nilgai and deer move large distances, nilgai specifically, and so we're trying to keep the cattle fever ticks isolated to these quarantine zones, but it's a difficult feat. So I'll kick it over to Sandra.
Okay, I'll take over from here just for a while. So South Texas is a great place to study fire because we can grow so much grass, and the grass is fuel. The grass carries the fire. The grasslands burn really well under prescribed fire conditions. In our part of the state, down there where that little circle is on the graph, in our part of the state, the natural or the historic return interval, I should say, was probably about 3 to 6 years. So it was fairly common. In addition to the grasslands, mixed in with the grass are some woody plants, some trees, and every once in a while there's a singlet tree just by itself, but usually they occur in a group, and we call that group a motte. The mottes are generally mesquite, live oak, huisache, and just like the rest of the grasslands throughout North America, woody plants have begun to encroach into the rangelands, and it changes the fire dynamics, of course. And so that this is what you see in this graphic from the University of Nebraska-Lincoln. And so one of the reasons to use fire in grasslands is to keep that woody encroachment at bay. Go ahead.
Here we go.
Our study site is El Sal's Ranch. El Sal's is in the Gulf Prairies and Marshes ecoregions. It's right along the coast. The eastern border of the ranch is the Gulf Coast in the northern part of the ranch. It belongs to the East Foundation, and East Foundation is an agricultural research organization. They manage the property and they research the property and are looking for answers to questions that landowners would have in South Texas in regards to both cattle and wildlife. So there are cattle on the ranch. It's just a very light to moderate stocking rate, which is probably about 1 animal per every 8 to 15 acres. And the ranch is dominated, the vegetation on the ranch is dominated by warm season grasses and forbs. The two main grasses are Gulf cord grass. And if you look in that photo there, the bunch grass, that big bunch grass, that's Gulf cord grass. That's a Spartina. And then there's also seacoast bluestem, which is just a variety of little bluestem. The little bluestem is probably the best forage grass for cattle. The Gulf cord grass is fair forage and wildlife value at best, because when it gets old, when it's an old plant, an old wolfy plant, and it's decadent, it's very coarse, it's very stiff, it's even pokey, like you want to wear chaps to walk through it because the ends are almost pokey like an agave plant. So a cow's got to be pretty hungry to want to eat that. And then of course there are those woody mott's that we talked about scattered throughout, and honey mesquite and live oak probably are the two most dominant woody plants in those. Go ahead. Our study design is a true patch burn grazing design. In other words, there are no interior fences. There's just fencing around the ranch on the perimeter of the ranch, but animals are able to go into the burn units, leave the burn units as they please. We don't even move them out for burning. So just like wildlife, the cattle can choose to be wherever they want to be. We've got 16 patches. The patches are a minimum of 500 acres. Maximum, I think the biggest one is like 1228 or something like that. We started these treatments in 2016, and we were just looking at the patches that are numbered 1 through 10 right along the eastern part, right along the coast. And then the ranch, the East Foundation wanted to expand, and we came in farther into the interior, and we added the 11 through 16 burn units. When we added those burn units, originally the study was summer versus winter burning, and then we also had control plots. When we added 11 through 16, we also added a return interval variable. So they might get burned every 3 years. They might get burned every 5 years. So we have summer short, summer long, winter short, winter long, and then controls that don't get burned at all. Go ahead. Just as a quick review, I want to tell you what we've already done since we've been doing this since 2016. Of course, we've been looking at herbaceous plant composition, and we've finished up some studies, but we always look at that because it has a lot to do with weather as well, and we're looking at different questions as we go. We looked at mortality of Gulf cordgrass because that Gulf cordgrass is, when you find it, a lot of times, it's almost like looking at a pure stand of Gulf cordgrass, and it looks like there's very little vegetation in there. Once we started studying it, we realized there was more in there than you could see, but it takes burning to really give those other species an opportunity to thrive. We looked at fire temperature, comparing season and comparing summer and winter seasons, and the nutritional quality of the herbaceous plants, and the movement of the livestock. How does the burning affect the density of the livestock and the distribution of the livestock? We have also completed a study on how does the burning in summer and burning in winter affect butterfly populations, because it's a very diverse region. It's the South Texas coast is the birdiest part of North America, and there are also maybe more butterfly species here than you would find throughout the rest of the country as well. So we looked at butterflies, and we have some ongoing studies right now on Texas tortoise, which is a state threatened species. And we're also looking at nearest neighbor relationships in the herbaceous plants. I'm going to turn it back over to Alynn to tell you about our tick protocol.
So the most recent project layered on Sandra's extensive prescribed burn work is our tick surveillance protocol and study. So what we started to do was survey for ticks in areas that had either been burned already in the past with Dr. Rideout's past burning and have recently been burned or have never been burned. So there's a mix of control and burn plots. And so what I've got here are asterisks on the plots that we're surveying in. So plots 1 and 5, those were burned in February of 2023, and we started surveying those plots in February, I believe, of 2023, as well as the control plot 3. This past winter, I guess, February 2024, we added plots 15, 13, and 12. So plots 15 and 13 were burned in February of 2024, and we added an additional control plot that was more interior to the ranch, just to have a control plot in both regions, and then these burn plots in both regions. Plot 1 and 5, those are short return interval burns, and plot 15 and 13 are long return interval burns. And so we are using the drag method, the 1 meter squared cloth, to collect ticks from the environment from these control and burn plots. So just a little infographic here. So we do drag two 750 meter transects through the grassland area or the non-motte area. And then within each plot, we survey the circumference of 3 to 4 mottes. And we mostly did this because being from the north, when I was surveying for ticks at places like Gettysburg National Battlefield, you could survey using the drag method in fields and you would get tons of amblyoma and maybe occasionally dermacenter. Down here, when we first started surveying, I really wasn't getting anything out in these tall grassy areas, and I thought, what is going on? And then Sandra and I started surveying around these woody mottes, and we were getting much higher tick abundances. So we thought, well, it's informative to have both of these in our study design because, of course, the grasslands burn better than the motts, as Sandra was saying. So each plot has 2 750 meter transects and 3 to 4 mott circumferences. And this is just our study, our plot locations again with those motts, which are the little like salmon-y colored plus signs, and our transects. Some of the motts seem like they're on the edge of the control plot. I'm thinking specifically plot 12. There's a lot of sand dunes on this ranch, and so we had to sort of finagle our sampling areas around those sand dunes and where motts actually are. So in some areas, in some of the plots, there are fewer motts than in others. So here's just a demonstration of a mott in the field unburned, which is on the left side. This comes from, I believe, our control plot on the coastline. And then here on the right is a photo of a post-prescribed fire, about 1 week post. You can see some of the vegetation just surrounding the mott did not necessarily get burned, although it looks like one of the trees in this photo did maybe catch fire. But that seems to be pretty typical, that the fire runs up to the mott but does not necessarily burn all the vegetation under or within. And if you have more questions about that, Sandra can speak to that much better than I can. I just wanted to show sort of our observations that we see down here. So when we do our drag surveillance, we check the drags every 10 to 15 meters, and all ticks are collected and stored in molecular grade ethanol, mostly because we want to be able to do pathogen diagnostics down the line, but also in case we pick up something that's visually difficult to identify, we want to be able to run polymerase chain reactions to identify the tick species using molecular data. And I'm sure everyone knows these are actually black-legged ticks in this vial. I think this was from Gettysburg National Park, so or National Battlefield. So this is actually an old photo. This is not from South Texas. We have yet to pick up a black-legged tick, but we know we've got Lyme here, so we're not sure what's going on with that. But as far as we know, we have not picked up one yet, at least at our site. And so our main research questions with this project, first we wanted to understand what the phenology of tick species were in South Texas. So we know emergence times in the north are pretty well set with emergence of nymphs before larvae, and then adults sort of peaking end of summer, early fall. And we wanted to know, does that hold true in South Texas? We don't really have distinct seasons here. We have about 2 weeks of winter, and then the rest of the year it's just hot. So we're kind of functioning under different dynamics here. And our animal hosts are pretty active year round. Our other questions were, do moths act as a great environmental reservoir for ticks, which, anecdotally, we already think yes. And the major question, do prescribed fires affect tick density? And if so, for how long following fire? And here's a picture of Rachel collecting ticks off a drag in I think this is one of our control plots. And so we're really only at the stage of starting to look at phenology. We haven't ID'd all of our tick species quite yet. And so we're just going to do some preliminary, we'll look at some of our preliminary data here and then talk about our next step. So this graph is just showing our tick densities of each life stage. So this is all tick species combined because we haven't ID'd everything yet. So all tick species just from our moths. So not our grassland transects, just our moth circumferences. And the surveys here start in March of 23 and they go through April of 24. We're still collecting, but this is all I've got on this graph for right now. We haven't done our May collections yet. And we've got larvae in the blue, nymphs in the red, and adults in the gray. And preliminarily, we need another year of data to really make any sort of concrete assessments here. And oh, sorry, I should say this is only using, I think this is only using the control plots. Actually, no, this is all plots combined. So that's why we have a lot of zeros on here. But in general, it looks like our nymphal peak is in the late-ish winter. So March to April, that's not too different from what we see in the north. I guess March would be a little bit earlier, but April, May, I think, for at least black-legged tick. But again, this is all species of tick combined, so this might tease out once we have our species identified. And preliminarily, we're not really sure what's going on with our larval peaks. It seems like there might be kind of a late winter, early spring peak in February or April, and then potentially a midsummer peak, maybe June or July. Again, we've only got 1 full year of data at this stage, so we're hoping to get at least 1 more to get us 1 year out from the burns that happened this past February. So we're going to keep looking at this and hopefully assess more clearly what our phenology is down here and tease out maybe species-specific dynamics. Here, we're just looking at the moth versus transect. So we've got moth in that top panel and transect in that bottom panel. Again, the life stage remains the same with larvae in blue, nymph in red, and adult in gray. And I just wanted to demonstrate how much the densities of ticks are different between these two sort of landscape features. So the grassland versus our woody moths. And I want to point out, I don't know if you can see my cursor, but our y-axes are different. So our y-axis on the transect is the max is much lower than the max on the moth. And so I just want to demonstrate that we do see much higher densities of all life stages in general in the moths, suggesting that these are an important feature for ticks in the landscape and may serve as environmental reservoirs, especially during prescribed fire, because they don't burn super well. And so this is just a breakout of our different plot type. So on the top panel there, we've got our control, which we've already seen. This is the control. This is just for the moths again. So we've got our control with the moths at the top panel, our long return interval fire, prescribed fire. I've got a red dotted line when the fire happened. So this, we have some preliminary, 1 month of preliminary data, and then the burn occurred, and then we've got our couple months so far post prescribed fire. And then at the bottom here, we've got our short return interval fire. That fire happened in February of 2023. So we've got a whole year of data following that prescribed fire. And again, the larval, nymphal, and adult colors remain the same. So again, this is just sort of a visual assessment of what's going on. We don't have any formal analyses here. What we thought was kind of interesting is that our nymphal peak and our short short interval prescribed fire post-fire was still about the same time as in our control plot. And again, the axes are different, so I just want to make note of that. But it doesn't seem at the moment that our nymphal densities are too much lower. Again, this is for the moths though, so we do still need to assess the transects, which I'll show you here. So I think this is just a little GIF. So it oscillates between our transect densities and our moth densities. It's that same figure with our control plots at the top, our long fire return interval in the middle, and our short fire return interval at the bottom. And I just wanted to demonstrate again the huge difference in densities of ticks of all life stages found in the moths versus in our transects. The y-axes are held constant for each plot type. So for control, it's held constant from moths to transects. Long is held constant from moths to transects, but they do differ between plot type. So this is just meant to be a visual representation of the differences in tick densities across our moths and our transects. That is all we have for our preliminary results right now, but we have some pretty ambitious next steps that Rachel's going to be leading. So first of all, we're going to get in there and identify all of our ticks, either morphologically using microscopy. That's likely the method we'll use for our nymphs and our adults. For the larvae, we'll probably use molecular ID. We're probably going to use a pooled system because we do have many more larvae than our other life stages, and so we'll probably pool them together and make some assumptions about when we get 400 larvae on a drag that they might all be from the same species, but we want to confirm that. Then we plan to do some pathogen screening. We're at least going to look at Anaplasma, Ehrlichia, Borrelia, and Rickettsia. We do have some limitations about which pathogens we can look at because we are on private ranch lands, and there are consequences for finding certain things like the babesia, which is again transmitted by the cattle fever ticks, which we don't even know if we have because we are outside of the quarantine zone, so we don't expect to find them where we are. But other tick species that may be carrying it, there are consequences for that. So we're still discussing whether or not the landholders would like us to look at that. And then we're hoping to do some formal analysis of this. We're probably going to do some modeling exercises looking at the different tick species and their densities, and specifically looking at time since burn, habitat type, local weather, and potential seasonal dynamics, and how those all influence both tick densities and pathogen prevalence in this system. And with that, if anyone has any questions or comments, or Sandra or Rachel, if I've missed anything, please feel free to jump in. And yeah, that's all we got. We'll take any questions.
So, Lynn, would you go back to the slide with the scorched tree on it with
Yes.
the pictures? I did a really bad job. I had one job and I blew it. I was supposed to describe how the moths don't burn. So let me just cover that real fast because I totally blew that. The grasses burn really well, like I said, but there's no grass under the trees in these moths. And so the fire just falls apart. It goes around the moth. And so that's why if you're a little bit confused about, why that doesn't burn, that photo there showing you what's left, it gets really weedy around the moth. So those are just forbs on the ground there, or weeds or wildflowers or whatever you want to call them. And that one tree just got scorched because the fire was hot, but the fire just goes right around it and it picks up on the other side. And so these are left as unburned refugia on the landscape. So I should have been more clear about that earlier. I apologize.
Thank you, Sandra, for pointing that out. I leave all the burning information up to you, Sandra, because for me, I don't know what's going on most of the time, but yeah, you're the expert.
Well, all I know about ticks is they bite, so we make a good team.
We do. We make a good team. But yeah, if anyone has any questions or suggestions or comments, maybe we've overlooking something, we're happy to hear that. Also, our contacts are on there, both Sandra's, myself, and Rachel's. So again, Rachel is the master student leading this project. Yeah, we'd be happy to hear from folks.
So Tammy posted a question. Do you know anything about rodent response to fire? Alynn, you went a couple of times with that team. There's another team from another university that was looking at rodent response. And I really don't know what they found yet that hadn't been communicated to me, but Alynn might know.
I do not know. So Tammy, I think there's a group out of College Station that was looking at recolonization of rodents in these prescribed burn areas. I don't think they've published anything on that. And I don't know if they're still surveying. I know they were out last spring surveying after Sandra burned those eastern those coastal plots, but I haven't seen them out this year, and I don't know if they're planning to come out, but I do think they were trying to look at recolonization of those plots by rodents. And I went out to pick some ticks off of some of their rodents. But yeah, I haven't heard much on that front.
While you're thinking of questions, and you can put your questions in the chat or like raise your hand and unmute and ask them. But Ryan Anderson, if you can put the post quiz question up, please.
Do I need to stop sharing for that, Ryan? Not sure.
I think you should be good.
Okay. Rachel, while they're taking the poll, did you have anything to add? Rachel is now spearheading this project. She's grabbed it and run with it. So she's going to be our resident expert.
Yeah, just about some of the gaps that we're finding in the literature. A lot of the researchers that did the tick collection studies post-fire weren't the actual burners. So I think like that's a big reason for a lot of the data on the burns being missing. So I think that our study will help provide really helpful information on different burn techniques and their effectiveness for tick control.
Awesome. Thanks, Rachel.
Thanks, Rachel. There's another question from Tahira. I hope I said your name right. Is there a hunting and conservation effort regarding wildlife species, parenthetically range expansion for cattle fever or cattle tick fever? So they do on East Foundation, they do hunt nilgai. Nilgai are very popular game species, so hunters come from all over the country to hunt nilgai there. I don't know that it's necessarily an effort to keep cattle fever tick at bay, but Alynn might have more information about that.
So on the east property specifically, we're not in the quarantine zone, and we technically don't have cattle fever tick documented there. But nilgai do persist there, and they do harvest them for commercial sales of the nilgai meat, which is meant to be very very popular and very tasty. In the south, in those fever tick zones, there's been a lot of methods, not necessarily hunting, but they've tried to put up fences and fences to keep the movement of nilgai at bay, and also basically like treatment areas where they would spray the nilgai with a topical treatment that would help remove the ticks or kill the ticks. Those have been really challenging. Nilgai are super, super neophobic. So any change to the landscape, any human-made intervention, they avoid. So, you know, if a nilgai are not good at jumping, they often cross under fences. So the higher the fence, it doesn't add any limit. It doesn't have extra protection for nilgai because they just go under. But if you try to put like one of those little treatment things over a spot where the nilgai are continually crossing under a fence line, they'll actually just avoid it after that. So I feel like there's been some efforts, not necessarily in the form of hunting, but some efforts to try to keep the tick at bay, but it's been very challenging just because of the nilgai behavior.
Yeah,
it's-
that study, there was a study, and it was led, I think, by John Goolsby, Goolsby, if you want to look up, it's probably published, but John Goolsby, and then one of our colleagues, Randy DeYoung, also worked on it, and there were other people that I'm probably leaving out, but they had set up sprayers, like Alynn said. Nilgai are very wary of any novel thing in their environment, so once the spray apparatus was set up around at fence crossings, and especially when they were near latrines, because nilgai will come and they will all use the restroom in the same spot. As soon as those sprayers went up, the nilgai stopped coming because they're just like, nope, I'm not going to go there anymore. So that's the only effort that I know of. It was not a hunting effort, like Alynn said. But if you want more information about that, I would search Goolsby, Goolsby, as the author on that.
Do
Thank
we
you,
have
Scott.
do we have any more questions? Oh, what materials are you using for your tick tricks?
We're using like a thick, it's kind of like a thick cottony material, almost a flannel, but not quite that heavy. We had a really nice like corduroy material that we were using in the Northeast, and I can't seem to find it, but it's kind of like a cotton flannel type deal, somewhere between not a full flannel and not strictly cotton.
Is it a muslin, Alynn, or no?
I'm not
Heavier than
sure.
that?
It's on the lighter side. It's on, so maybe it is that. I'm not sure. It's what I picked it up from Hobby Lobby. Old faithful Hobby Lobby.
Yep.
And we're using an aluminum camping pole. Okay. Yeah, I'm not sure if it is that material or not. What's your experience been with that?
I'll just come off and mute right quick so I don't have to type super fast. We've been sampling for questing Diabopictus larvae for this will be the third year now, to see if there are any hot spots where the moose might go in the spring during tick drop, and then in the fall when the larvae are questing. And it just seems like we're not trying to get population density, so maybe it doesn't matter as much. We've been trying to compare random transects with game trails around certain areas, and I'm just wondering if the larvae stick to the muslin as well as they could to some other thing like flannel. So, something fuzzier.
Yeah, our sheets they have that fuzzy look. That's why they're like somewhere in between, like a cotton blend and but they're not as thick as a flannel, which is unfortunate because everything down here is really thorny, but they do have that like fuzzy bit. The ticks do seem to get caught in them, which has been useful. But yeah, there was a group I was working with that at the CDC. They had the best material. It was like this really thick, fuzzy, flannelly material, but I can't seem to find that. So, yeah, we might be missing some with our material, but I'm hoping it does have that fuzzy complexion, I guess. I don't know if that's the right word. Complexion. I saw someone said they were using waterproof bed sheets. That's a great idea.
Yeah, what are they made of? What are those sheets made of? Are they cotton?
They linked to them. Let's see.
Hi, this is Leanne. Sorry, my voice is not that great. But yeah, we use I found these waterproof bed sheets. They're like a fuzzy flannel on one side, and then they're kind of plastic coated on the other side. So it helps, like, then when we flip the sheet over to collect the ticks, then both sides of the sheet don't get all full of burrs and twigs and leaves and things. So they've worked out really well for us.
Oh, that's a great idea. We do have lots of again, little spiky seeds down here that get caught up in the
Mhm.
drag and on your knees when you are down looking at the drag.
Yeah, that's the same with us. So so I really like these that are more of just like the plasticky surface on the one side, and then they are fuzzy flannel on the other.
Thank you for sharing that. I appreciate that.
Mhm.
Oh, and the denim fabric from Joann's. I think that's what I was using with the CDC folks. Yeah, copy. Is there a way, Ryan, that we could get a copy of the meeting chat, or do we just have to like copy and paste it? The discussion of the materials.
What happens if I hit save chat?
Oh, that could work.
Yes,
yeah.
I just like downloaded a file. I can send that to you.
Okay.
Unless you also have the ability to save chat. I don't know what the permissions are.
I think I can. Oh yeah, yep. I was able to save it as well.
Perfect. Yeah, does anyone else have questions or experience to share on tick collection metrics and materials? Suffolk County, New York, we use wide whale corduroy.
Ooh. I'm not sure if I even know what that is, but it sounds good.
I think that's just the corduroy with the big stripes instead of the tiny.
Oh, okay. Okay.
Correct me if I'm wrong, Scott.
Yes, that's correct. Yeah, it's the one with the wide whale has the larger strips.
Okay.
It's hard to find, but it really is durable when it comes to, cat briar and getting hung up on things like that. It doesn't tear very easily. So we found it very useful up here.
Thank you for sharing that. That would be very useful down here as well. All of our mesquites are really thorny, and we've actually surprisingly not gone through too many drags yet, but I've torn a big hole in one of them. I don't know how Rachel's been doing, but yeah, they do get torn up quite a bit.
We have several acacia species too, and 3 or 4 things that get called cat claw just because that's what they look like. So
And feel like.
yes,
Oof.
that's what they feel like.
Here in New York, when we make we flag, we don't drag, but when we make the flags, we actually do it two-sided, because the one side of the corduroy is very flat, it doesn't have much plush. So we actually put those two sides together on the interior. So the exterior wide whale plushy form of the fabric is on the outside. So it makes it a little heavier, but it's very sturdy and doesn't really tear as much as flannel or something on a lighter variety.
Awesome, thank you. I don't know if there are any other questions, comments.
I do want to note just once again, our emails of our presenters are on the screen here. So please make sure to take note of those if you come up with other questions in the near future here. I will also put my email in again. If you have working group questions or anything of that variety. please direct them my way. So thank you very much, Dr. Martin and Dr. Rideout-Hanzak and Rachel. And thank you to all of you for being here today. This was very interesting, and we've had a really nice discussion here at the end. So I hope to see you all on June 12th at the same time. And in the meantime, have a great month.
Thank you to everyone who tuned in.
Yeah, thank you.
And thank you, Ryan, for hosting.
Yes, thank you.
Bye-bye.
Bye.